Welcome back again in a new video session from Biology Boost Up. And in this video session, we're going to study about the nucleic acid. So now let's start. The nucleic acid are non-protein nitrogenous substances transmitting hereditary character and directing protein synthesis. The nucleic acids are polymer of nucleotides. A nucleotide is made up of a nitrogenous base, a pentose sugar, and one or more phosphate group. The nucleic acid contain either a purine or pyrimidine as the nitrogen base and are accordingly called purine and pyrimidine nucleotides. Nucleotides contain either ribose sugar or 2-deoxyribose sugar as the pentose and are accordingly known as ribonucleotides or deoxyribonucleotides. The DNA or deoxyribonucleic acid are polymer of purine and pyrimidine deoxyribonucleotides, whereas the RNA or ribonucleic acid are polymers of purine and pyrimidine ribonucleotides. Now look the components of nucleic acid. First, the nitrogen base. It could be purine or pyrimidine. Second is pentose sugar. It also may be ribose or deoxyribose sugar. Next, the phosphate group, that is phosphoric acid. The chemical structure of purine group and pyrimidine group. First, in case of purine group, it is larger than pyrimidine molecule. The pyrimidine molecule is smaller than purine molecule. The purine is 9-membered molecule, whereas the pyrimidine is 6-membered molecule. In case of purine group, it contains of 2 rings joined at 4 and 5 position, but in case of pyrimidine, its molecule is a single ring. Next, the purine has nitrogen atom at 1, 3, 7 and 9 position. In case of pyrimidine group, it has nitrogen atom at 1 and 3 position. This is the chemical structure of purine. As you can see here, the purine group consist of 2 chiral rings, and those rings are joined at the 4 and 5 position with each other, and here 4 nitrogen atoms also present at the position of 1, 3, 9 and 7 of the chiral rings. This is the chemical structure of pyrimidine group, and it is a single ring molecule. It has 2 nitrogen atoms at the position of 1 and 3. Now next, the types of purine and pyrimidine base. Basically, the purine are 2 types. One, adenine or 6-amino purine. Second is guanine or 2-amino 6-oxypurine. And the pyrimidine base are three types. First, the cytosine or 2-oxy-4-amino-pyrimidine. Second, thymine or 2,4-dioxy-5-methyl-pyrimidine. And third, the uracil or 2,4-dioxy-pyrimidine. Now, let's talk about the structure of sugar molecule. The sugar molecule in a nucleotide may be ribose sugar, that is C5H10O5, or deoxyribose sugar, that is C5H10O4. Both sugars are in furanose or pentagon state with 4 carbon and 1 oxygen atom. The fifth carbon along with H2OH occurs outside the ring. The deoxyribose sugar has 1 less oxygen at 2' carbon compared to ribose, that is deoxygenated condition, and thus named so. In other words, ribose molecule differ from deoxyribose molecule in having a hydroxyl group instead of hydrogen at carbon number 2. Now look at the structure of phosphoric acid or H3PO4. It is biologically called phosphate group because it dissociates under physiological conditions, releasing hydrogen ions. A nucleotide may have 1, 2, or 3 phosphate groups, but in nucleic acid composition, only nucleotides with 1 phosphate group are involved. Next, what is phosphodiester bond? A phosphodiester bond occurs when exactly 2 of the hydroxyl group in phosphoric acid react with hydroxyl group on other molecule to form 2 ester bonds. An example is found in the linking of 2 pentose ring to a phosphate group by strong covalent ester bonds. Each ester bond is formed by a condensation reaction in which water is lost. This bond is a key structural features of the backbone of DNA and RNA and links the 3' carbon of one nucleotide to the 5' carbon of another to produce the strand of DNA and RNA. Next, the process of phosphodiester bond formation. In phosphodiester bond formation, 2 hydroxyl groups on the phosphate molecule bind to the 3' and 5' carbons on 2 independent pentose sugar. Those are 2 condensation reactions. So 2 molecules of water are produced. The phosphate is then bonded to the sugars by 2 ester bonds. Hence, the nomenclature of phosphodiester bond. This reaction is catalyzed by ligase, such as DNA ligase during DNA replication. This is the process of phosphodiester bond formation. Thanks for watching my video. Thank you, friends.